Hi everybody, my project is about genotyping for genetic disorders. To do this, a scientist identified an SNP, or a single nucleotide polymorphism. In other words, this is a mutation in the DNA. For example, a thymine might be changed to a cytosine, or a guanine to an adenine, or any way around. Now, this mutation can cause either a huge effect in the phenotype and can change a lot of things, or it can have a small effect on the phenotype and only change a few things. With that being said, the mutation can be a good thing or a bad thing. It can either produce a good outcome or a bad outcome, depending on what it is. The mutations can happen in DNA or they can happen in RNA. To identify an SNP, special probes have been designed that can identify a specific sequence of DNA. This specific sequence of DNA is the SNP. If the SNP is identified, the DNA will stick to the probe in a special way, and by looking at it, the scientists can determine whether it's the SNP or not the SNP. Now, SNPs are the basis of some genetic disorders. Now, this means that some genetic disorders that are caused by a certain sequence of nucleotides, this SNP can mutate into that certain sequence and cause the disease. This can also be inherited from parents. Now, this can help with medicines. If the scientists know the SNP, they can create medicines that might counter it. For example, viruses will do that. A virus will replace with the plasmid, and it'll work. It'll change the DNA sequence and take out the genetic disorder. On the other hand, livestock can be breeded with a good SNP. So if a cow, for example, has a good milk gene, maybe that's an SNP. So they'll breed the cow with hopes of the offspring having that SNP. The reason I chose this topic is because I myself have a genetic disorder. The disorder is called junctional ectopic tachycardia, or JET for short. JET is normally found in people who have had a heart surgery, and it's just kind of like a post-operation temporary disorder that they'll get, and it'll go away within a few days. Mine's a little different. I was born with my disease. Now, researchers don't know much about the disease. I don't think they know what chromosome it's located on, and I'm not sure if they know the SNP. With that being said, I've made my predictions. First prediction is that it could be an autosomal dominant. The reason I say autosomal is because both my sister and I have it, so it can't be sex-linked. The reason I say dominant is because it's so rare, it's a very slim chance that my mom and my dad would have a recessive gene to give to me. So, I think that it's a dominant gene that came from my dad. Now, I also think it could be a mitochondrial disease. Considering the fact that it came from my dad down to my sister and me, I think that it could be mitochondrial. Then again, the chances of it being dominant and just a 50-50 could have played in there, but I also think it could be autosomal. Alright, here's the bibliography.